Hi, today we're going to be talking about neural communication. So neural communication takes place in nerve and muscle cells, which are excitable tissues. So this means they can rapidly alter their membrane permeabilities and undergo membrane potential changes when excited. So we call these changes in potential the electrical signals. So compared to resting potential, a membrane becomes depolarized when the size of its negative potential is reduced, becoming less positive, and hyperpolarized when the magnitude of its negative potential is increased, becoming more negative. Changes in its potential are brought about by triggering events that alter membrane permeability, in turn leading to changes in ion movement across the membrane. So the two kinds of potential change are graded potentials that are short distance signals and action potentials, the long distance signals. So let's go ahead and let's take a look at our slide, which is the structure of a neuron. On the left, you have the cell body, which is the life support system. It is surrounded by the dendrites that receive the messages from the other cells. The signal or the nerve impulse then goes down the axon to the terminal branches that you'll see on the right-hand side. If they have a myelin sheath on them, that helps to speed up the nerve impulses. Okay, now let's look at a slide that has a couple of different neurons, and the action potential shoots down the axon away from the cell. That happens here from 1 to 2. And at 2, a neurotransmitter is released into the synapse where the dendrites of the neighboring neurons detect it. So that's how they connect together. If there's a receptor for the neurotransmitter on the dendrites, the neurotransmitter and the receptor bind, creating an electrochemical signal. And then if that signal is strong enough, it will then spread down the dendrites and across the cell body of the next neuron, and it begins another action potential. Okay, so next let's look at the graded potentials, which is just merely a local change in membrane potential that occurs in varying grades of magnitude. It serves as a short distance signal in excitable tissues. So if you look at the diagram to the left, on the top, you'll see the resting membrane potential. This is the hyperpolarizing graded potential. You notice how it dips down on the bottom. The depolarizing graded potential is below that, and you'll see where that comes up in the green. So if we wanted to talk about potential membrane changes, if you had a membrane and it changed from a negative 70 millivolts to a negative 60, we would call that a 10 millivolt graded potential. If we had one at a negative 70 and it changed to a negative 50 millivolts, that would be a 20 millivolt graded potential. So a graded potential, usually a depolarization, occurs in a small specialized region of an excitable cell membrane. So the site undergoing a potential change is called an active area, which is what you see in the green or the blue on that slide. So graded potentials spread by local current flow between the active area and the adjacent inactive areas, and they die out over short distances. The magnitude of a graded potential varies directly with the magnitude of the triggering event. So a stouter triggering event, the larger the secondary graded potential would be. So graded potentials will spread by a passive current flow, and they will potentially die out over short distances. Next, let's look at the action potential. And let's just start with the slide. If you will notice on the left-hand side, as it gradually starts to go up, that's your local potential change, which is the graded potential. And then as you see it spike directly up, that's the depolarization, the opening of the voltage-gated sodium channels. Once it makes it to the top, and then it starts to come down, that's your repolarization. And what happens there is the closure of the sodium and the opening of the potassium voltage-gated channels. Down at the bottom, you'll see where hyperpolarization takes place because those voltage-gated potassium channels remain open after the potential reaches resting level. So your action potentials are propagated from your axon hillock to the axon terminals. And once initiated, action potentials are conducted throughout your nerve fiber. And the refractory period ensures one-way propagation of action potentials, and it limits their frequency. So action potentials occur in an all or none fashion. And the strength of a stimulus is coded by the frequency of the action potentials. Myelination, myelination that we talked about on the axon earlier, it increases the speed of conduction of action potentials. And the fiber diameter also influences the velocity of action potential propagation. Okay, so next, let's just take a real quick summary and let's look at the differences between graded potentials and action potentials. On your graded potentials, they're going to vary in magnitude. They're going to vary in duration. They're going to decay with distance. They occur in the dendrites and the cell bodies, and they're caused by openings and closings of many kinds of ion channels. Whereas in the action potentials, they're always the same magnitude in a given cell type. They're always the same duration in a given cell type, and they can be transmitted across long distances. They occur in the axons, and they're caused by opening and closing of voltage-gated ion channels. So the refractory period is the time when a recently activated patch of membrane is unresponsive to any further stimulation, and this prevents the action potential from spreading backwards into the area that it just passed. It ensures unidirectional propagation of the action potential away from the site of activation. So if you were doing an experiment with a neuron and you could stimulate both ends of it simultaneously, they would actually meet in the middle and stop. Once they move towards each other, they would stop in the middle of the axon. The two adjacent patches of the membrane in the middle would be in a refractory period. So further propagation of either action potential would be impossible. It's impossible to stimulate the portion of the membrane where the pulse has just passed until it's recovered from its refractory period, ensuring the one-way propagation of action potentials. Okay, the structure and the function of a single synapse. In the nervous system, a synapse is a structure that permits a neuron or a nerve cell to pass an electrical or a chemical signal to another neuron. In the past, you may have heard this referred to as a junction. It's just an older term that means the same thing. One neuron directly interacts with another neuron primarily through a chemical synapse. Most neurons have 4 different functional parts. You've got the dendrite cell body region, which is your input zone, and it serves as a postsynaptic component that binds with other response to neurotransmitters released from other neurons. And then you have the axon hillock, the trigger zone, and it's where action potentials are initiated because it has the lowest threshold, and it reaches threshold first in response to an excitatory graded potential change. Then the axon and the nerve fiber, which is your conducting zone, they conduct action potentials in undiminished fashion from the axon hillock to the axon terminals. Then the axon terminals are the output zone, and they serve as a presynaptic component releasing a neurotransmitter that influences other postsynaptic cells in a response to action potential propagation down the axon. The terms presynaptic and postsynaptic refer only to a single synapse. Most neurons are presynaptic to one group of neurons and postsynaptic to another group. The events that occur at a single synapse result in either an EPSP, excitatory postsynaptic potential, or an IPSP, inhibitory postsynaptic potential at the postsynaptic neuron. So your EPSPs and IPSPs are graded potentials. Unlike action potentials, which behave according to the all or none law, graded potentials can be of varying magnitude. They have no refractory, inhibitory, postsynaptic potential. All hyperpolarization of the postsynaptic membrane in response to neurotransmitter binding, thereby moving the membrane further from the threshold. So just make a note that your EPSPs and your IPSPs are produced by openings of chemically gated channels, unlike your action potentials, which are produced by voltage gated channels. So many different molecules serve as neurotransmitters, even though neurotransmitters vary from synapse to synapse. The same neurotransmitter is always released at a synapse. So at a given synapse, binding of a neurotransmitter with its appropriate subsynaptic receptor channels always leads to the same change in permeability and resultant change in potential of the postsynaptic membrane. So the response to a given neurotransmitter receptor combination is always the same. The combination does not generate an EPSP under one circumstance and an IPSP underneath another one. So let's just look at some common everyday examples. So we know that pain is what protects our body from damage. So years ago, I burned the left side of my leg when I leaned into a hot pipe on a motorcycle, and I went to ER for a third-degree burn. My leg could be pulled away from that hot pipe. It happened so fast that I can't recall exactly what muscles pulled away, but the summation of EPSPs at the cell bodies of the neurons controlling the muscles brought the neurons to threshold. And the subsequent action potential that was generated in these neurons would stimulate a contraction of the muscles. So I can tell you that happens very quickly. I've also had my blood drawn, as I'm sure many of you have. And even though you know a prick is coming, you could hold your arm out still for the prick, so that simultaneous contraction of the triceps muscle, which would oppose the flexion of the elbow, would be prevented by the generation of IPSPs at the cell bodies of the neurons controlling the muscles. So these IPSPs would keep the tricep neurons from reaching threshold and firing so that they would not contract. So the arm can be extended, even though that there will be pain, voluntarily generating EPSPs, which override the IPSPs at the bicep. So next week is the central nervous system. Thank you.